joining us this afternoon on the last day of Psych. For those of you who are new here, Science Gallery Bengaluru is a public institution for research-based engagement, and Psych is our 5th exhibition season, the 3rd, which is completely digital and online. Over the past 45 days, Psych has been unraveling the complexities of the human mind. And today, as a part of this exhibition season, we're very pleased to bring to you the walkthrough titled Disease to Dish, a closer look at the Accelerator Program for discovery in brain disorders using stem cells. Before I tell you a bit more about today's event and all the experts we have with us, I'd like to tell you a bit more about what's coming up this evening on the last day of programming at Psych. Do join us after this event for a discussion between filmmaker Alec Widdowson and sociologist Gareth Thomas about Alex's film, Music and Clowns. This is also the last day to watch the film, so please do have a look at that. And join us later in the evening for the closing event, which is a lecture by psychiatrist Vikram Patel titled Reimagining the Path to Recovery, a reimagination of the mental health care system in India. Now I'd like to introduce you to Biju Vishwanath, who will be leading today's session. Biju is an additional professor of psychiatry at NIMHANS. He is a physician scientist with a PhD in the area of cellular models for psychiatry. He has recently been awarded an intermediate fellowship of the DBT Welcome India Alliance and is the coordinating investigator of the ADBS program, that is the Accelerator Program for discovery in brain disorders using stem cells. His areas of interest include the biological basis of drug response in psychiatry, especially lithium and bipolar disorder. Along with Biju, we have several other experts who are there today in the panel, including Dr. Sanjeev Jain, Dr. Ritika Sood, Dr. Meera Purushottam, Anushka Banerjee, Yojit Sharma, and others. So Biju will introduce everyone to you as a part of this event. I'll also like to remind the audience that there will be an interactive Q&A session at the end of the event. So do keep your questions in hand and type them in the Q&A box. The team will be very happy to answer them at the end of the session. You can also share your thoughts with us about today's event, about the exhibition season, and what other kinds of programming you'd like to see from Science Gallery Bangalore in our feedback form. The link is in the chat box. Now, without further ado, I'll hand it over to Biju.
Thank you, Madhu. I'll just share screen. My screen visible?
Yes.
My screen visible? Yeah, thank you. Good afternoon. So it's a pleasure to be part of this program. Disease to Dish, a close look at the ADBS. So we'll jump right away to the topic. So here, what we know very well is that the burden of mental illness is huge, both globally and in this country. So 1 in 7 Indians is affected by a mental disorder. So this is amounting to 200 million individuals. And this is not just a number, 15% of total lived years with disability. So the common severe mental illnesses like schizophrenia, bipolar disorder, obsessive compulsive disorder, and other anxiety disorders, these constitute most of this. This leads not just to disability, but also early mortality. People die because of suicide due to medical causes which are unreasoned because there's so much stigma about mental illness. The most important reason for all of this is that the biological basis of these mental illnesses is mostly unknown. And although there are treatments which help, people hesitate to take them, but then there are no disease-modifying treatments. They only help with symptoms. So when we started the program, ADBS, it was primarily meant to tackle some of these questions. So the ADBS, or the Accelerator Program for Discovery in Brain Disorders Stem Cells, is a multi-institutional program. It includes NIMHANS, NCBS, and INSTEM from Bangalore, and CIRA from Kyoto in Japan. And as you can see, there are so many investigators who are very eminent scientists, and I've been lucky to be part of this group. This is funded by the DBT, the Pratiksha Trust, and essentially looks at a clinical cohort of families, so a large cohort of families who have these mental illnesses, and to try and analyze altered brain function. So setting up the cohort and following up this cohort, do detailed clinical assessment, and then try and do assessment of brain measures, try to generate cell lines from these patients, and then try to do functional studies with them to try and understand the fundamental brain mechanism. So what we are trying to do here is that I will try to, what we want to do was to do a live video, but it was such a big program with so many institutions, so many labs, which all come together. And so what we are trying to do is to have a pre-recorded video of the various parts of this program. So we'll go towards that. Madhu and Gayatri.
Thanks, Biju. If you can stop sharing your screen. Thank you.
Hi, my name is Srinivas and I'm a psychiatrist working at NIMHANS for the ADBS. So I will take you through the clinical side of what we do in the ADBS study and how we assess patients and their families. So we approach patients from the outpatient clinics or from the inpatient wards, and so far more than 500 families have participated in our study. So this lovely building that you see is the Department of Psychiatry at NIMHANS. Let's go inside and see how we do these assessments. So this is the first step in the ADBS. When we come across a family who we think we can recruit as part of the study, we usually draw a pedigree and analyze them. So can you tell us what you're doing here?
Yeah, so here we are drawing a brief family tree or a pedigree of the participants of our study, where here we can see that the index participant is a proband of the diagnosis of bipolar disorder. Yeah. We also have his sister who's unaffected, and we can see that in this family, details of his family, there are multiple family members, like his father is also a diagnosed case of bipolar disorder, and many of his paternal uncles and aunts have been diagnosed with bipolar disorder, substance use disorder and psychosis, and we have his grandparents who also has a diagnosis of dementia.
Okay. So this is our first step, and we do see many families have several generations of people who have been affected, but at the same time there are quite a few people who don't have the illness as well.
So I have a question. So in my generation, actually I am the only person who is having a bipolar disorder as a mental illness, whereas my brother and my sister who are both are healthy and they are doing well in life. So why it is that?
Yeah, that's exactly what we are trying to answer through this study. So, and this is why we are trying to study both you as well as your sibling, your sister who does not have the illness, right? So we look at how your genes are different, your brain structure or brain function, what are the differences between you and her. And we'll also be asking you questions about your life experiences and things you have been through, whether any of them could have put you at risk for developing this illness.
Okay.
So now we'll be showing you how we assess an individual subject. So can you tell us what you're doing?
Yeah, so I have the subject with us who has been diagnosed with bipolar disorder. He suffered multiple episodes of what we refer to as mania as well as depression. So now, Alan, I'm just going to ask you a few questions about the most recent episode that you had, which was 3 years ago in 2019. So can you just tell me what symptoms you had at that time?
Yeah, so from what I could remember at that time, it was very difficult for me to get some sleep. Like I felt like I did not need to sleep at all. My energy levels, I felt like were off the charts. I was always running around and talking to people, even at night, I think. And then there was a long spree where I would, buy a lot of gadgets and, use it for a week, give it away to somebody, then buy something again and again. And yeah, that's the thing I could remember.
Can you tell me predominantly how you felt? Were you sad? Were you happy? Were you angry? Most of the time, how did you feel?
No, I was very happy. I mean, I don't even know why. Like, I thought the world was very positive and beaming, and that, it would be like that forever.
Right. During this time, did you also feel like you had a lot of special powers? Or did you feel very special about yourself?
Yeah, I don't think I felt like I had some special powers, but I felt like, I could do anything. Like, if I had the will to do it, it was possible.
And how long did this period last?
Yeah, this went for at least a month.
Right. And I also got this additional information from your file that you have had similar episodes in the past as well.
At least twice. There have been something, maybe a faint memory of similar episode maybe earlier, but I don't think it was as strong as what was reported during this time.
Right. And also in between, have you ever had a period where you've been really low? Do you recall a period when you've been really low and sad?
Yes, there was. Initially, I think even we at home, we not pick it up that easily because I normally tend to also keep it home otherwise. But once when I also noticed that I did not feel hungry anymore. I didn't feel like I have to take in food. I did not have any interest in keeping in touch with my friends, and it was very difficult to get my work done. Like my classes suffered. Like I couldn't do any of my assignments at home.
Right. And your predominant mood at this time, how would you describe it?
I can't say it. I mean, it was mostly sad.
Mostly sad.
It felt like there was everything, whatever I saw.
Right. And was there any point where you felt that you were so sad or so low that sometimes you felt life was not worth living?
Yes.
Yeah, can you just elaborate a little more on that?
Just like I said, everything that I saw through it felt like, what is the point of this? Why am I going through this? Is it worth, living a life like this at all? My parents got a little worried because I would talk about this once in a while, not every time, but when I couldn't hold it at all, I wouldn't cry, but I would tell my mother, I'm feeling things like this. So it quite worried them. They asked me, did I do anything stupid? That was the first thing. I did not understand what they meant by then at that time, but later on I felt I did not want to do anything severe, but I was really not feeling like this is how I have to live this life.
Right. So, and you began treatment only to take your treatment?
Yes.
Okay. Yeah. Thank you so much, Alan, for this information.
Inside, my colleague Dr. Soumya will explain to you how cognitive functions are tested.
Hello all. Now that you have seen the assessment about an family, the pedigree there, and an individual patient, how the symptom assessment and the live charting has been done, now we'll move ahead to something called as what is neuropsych assessment. In simple terms, it's nothing but assessing about the basic functions of the brain like attention, concentration, memory. So we'll go ahead with how a neuropsych assessment is done for the individual subjects.
Okay, I'm going to assess one subdomain of this neuropsych assessment, that is verbal learning memory. The test name is Auditory Verbal Learning Memory. So I'm going to call out a list of simple words. Listen to them carefully. Once I am done with the list, you will have to recall the words. While recalling the words, the order of recalling could be any way. The first word could come at the last or in the middle. But you'll have to recall the words from the list.
Okay.
Shall we begin?
Okay, sure.
Arm, cat, axe, bed, plane, ear, dog, hammer, chair, car, eye, horse, knife, clock, bike. Please tell me.
Arm, car, chair, dog, knife, bed, hammer.
Do you remember any more words?
Not that.
Is this all you can recall?
Yes.
Okay. I'll repeat the list. You'll have to listen to them carefully. So this test is called the Color Trails 2. It is part of a neuropsychological assessment, and here we are assessing some of the brain functions which are called cognitive functions, and they are related to attention, concentration, and this specific test is related to some functions like the sustained attention and cognitive flexibility. So now I will be starting the test. So hi Joanne. So here are numbers from 1 to 8, okay, and they are in both colors, pink as well as yellow. So what you need to do is you need to join these circles in a sequence, in the number sequence from 1 to 8, but you have to alternate between the colors. So if 1 is in red, 2 has to be in yellow, 3 red, 4 yellow, and you have to do it as fast as you can. When I say start, you start. Start.
Hello everyone, I am Dr. Samya, working as clinician scientist and assistant professor in Department of Psychiatry under the ADBS project. Now moving on to the next one, which are called as the endophenotypic markers. When I'm talking about the endophenotypes, if I have to put it in simple words, they are markers which are not only present in the patients with psychiatric disorders but also the unaffected relatives. And when I'm saying they are present in unaffected relatives, they are heritable and they are found in more with these families which have the members with multiple members with affected with psychiatric illnesses. And even in the patients, these markers, they are not only present in the affected state or the disease related state, but they are also present during the other times, which we call it as trait based markers rather than state based markers. So in this lab, as part of the ADBS, we will be looking at these two measures, which are one FNIRS, the functional near infrared spectroscopy, and the other measure is eye tracking. So when we are looking at first functional near infrared spectroscopy, this is a measure which detects the changes in oxyhemoglobin versus and the deoxyhemoglobin, the oxygenated hemoglobin and the deoxygenated hemoglobin molecules in the brain, which would in turn reflect the underlying brain activity. So this can be studied in rest as well as task related conditions, and since we are looking at the changes in their concentration, which is in turn reflecting the underlying brain activity, this principle is something called as neurovascular coupling or the hemodynamic response. This is something similar which you would have heard in functional MRI. However, this technique is more a simpler one, which will be able to tell us the underlying brain activity without going into an MRI. So the FNIRS, as I had mentioned, it is based on this principle of spectroscopy. There's a light source which emits quasi-infrared light at the scalp, and there's half of the waves which are being absorbed by these oxyhemoglobin or the deoxyhemoglobin which are present in the blood vessels of the brain, and a detector at the other end would absorb or would detect the light which is returning, and that would be a result of the interaction between these chromophores, that is oxyhemoglobin and deoxyhemoglobin. And you can see that the path is being like a banana shaped curve, and so the interaction between these oxyhemoglobin and the deoxyhemoglobin and the change in the concentration of these, that is what being detected, and which would reflect the underlying brain activity as well. So now we'll go ahead and look at how the procedure of FNIRS is being done.
Okay, so I'll make you to wear this cap. After wearing this cap, try not to move your hands and legs. Okay. You have to be relaxed, keep your eyes open for 5 minutes, and you have to look front. Okay. Okay? Okay. Stop. You're comfortable now?
Yeah.
If you're feeling any difficulty, let me know.
Yes.
Okay. Now focus straight and look front. Okay. So this detects the activation of the brain during rest.
The other endophenotypic marker which we are studying in the lab is the eye tracking measure. The eye tracking system is something which involves the infrared camera, which picks up the eye behavior gaze, like how the eyes are moving and how is it different in each of our psychiatric conditions. That's what we'll be studying. So when we are looking at the eye tracking abnormalities, as I had mentioned, it's one of the endophenotypic marker, meaning to say that it's not only found in patients with psychiatric disorders but also in the unaffected relatives as well. So among the psychiatric illnesses, there have been various illnesses among which the eye tracking abnormalities have been studied, most commonly in schizophrenia and bipolar affective disorder. So we'll look at the procedure of how the eye tracking measure is being done. We'll be able to analyze various measures like position measures, the movement or the velocity, and as well as the latency measures. So we'll have a brief look at how the eye tracking procedure is being done. We'll be having a brief look at the eye tracking procedure now. So before starting the actual procedure, we'll be checking for the eye dominance. First with both eyes, then the left eye is being checked for, then the right eye is being checked for. So the subject's dominant eye seems to be right eye.
This is the infrared camera which is focusing on the dominant eyes. Okay, now I'll start with the fixation. That is the first task. Now you have to focus on one point, and after some time one more point will come, but you have to look only at the center one. Okay. Now one point will come in the center of the screen and it will start moving. You have to follow the point. Okay. Close your eyes. Open your eyes. Look at the point.
So now that you have seen the eye tracking procedure, the last part you were able to see how the dominant eye is moving with respect to the target. And so in this you will be able to see that the black line represents how the subject's eye is moving, whereas the green line is the target. So we will be able to analyze how is the individual subject's eye behavior, which is different from that of the target. So green being the target, the black being the of that of the subject. So the deviance you are seeing, so that is what we will be measuring, mainly in terms of how the position difference is happening, as well as how the speed and the latency difference is there when compared to that of the target.
This is the Center for Brain Mapping Laboratory. Brain mapping means measuring the electrical activity either functionally or structurally or combining both. Many techniques involved in the brain mapping. One of the harmless, inexpensive technique is electroencephalogram. EEG is used to diagnose many number of conditions affecting the brain. There are different kind of EEG recordings. One is the routine EEG, which is mainly used for the diagnostic purpose. The other one is the research study, which includes a high density electrode array. In our CBM lab, we are doing both clinical as well as research studies. We can record the brain waves with the help of electrodes or wires placed on the scalp. Two different kind of electrodes available. One is individual electrode, which is mainly used for diagnostic purpose, example ring electrodes and cup electrodes. And the other is cap based, and this is useful for research studies. Example, the plain cap without electrodes, and the other one is sensor net, which is inbuilt electrode connection. We have 32 channel as well as 128 channel sensor net. As you can see here is a 32 channel sensor net, and this is the 128 channel sensor net, which is a high density electrode. We are using 128 channel EEG, which includes even facial electrodes to capture eye movements. you can see the solution, and we need to dip the EEG cap inside this for 5 minutes. After that, we can place it on the subject head appropriately. Earphones are connected on the subject's ear for the auditory stimulus. This is geodesic photogrammetry system, GPS. Subject can sit in the GPS equipment in a proper position. We need to capture multiple images from 11 cameras for locating the exact position of each electrode. It is helping in imposing the EEG for better structural position along with the functional measurement. EEG is one of the endophenotypic measure in the ADBS research project. Here we are recording the EEG for eyes open 1 minute and eyes closed for 1 minute. Same will alternate. While we record the EEG, we can give any kind of stimulus to the patient and we can record the event related potentials. Here we have given the paired auditory stimulus and EEG is recorded. EEG is a neurophysiological marker to identify different physiological and pathological conditions. As you can see here, this is dementia patients, schizophrenia and the healthy controls. All 128 electrodes measurements are here. You can see the reduced power in the patients when compared to the healthy controls. Example, in dementia, it is helping in predicting the stages of dementia. The early detection of dementia will help in the treatment as well as delaying the few representative scalp topography and the statistical analysis pictures you can see here. While we record the EEG, we can give any kind of stimulus to the patient and we can record event related potentials, usually called ERP. Here you can hear example auditory stimulus. When we hear paired sound, our brain response to second sound will reduce when compared to the first sound, the healthy control. Whereas in patients will continue even for the second sound. As you can see here, the peak-to-peak amplitude reduction is seen in the schizophrenia group than the controls. P50 auditory ERP is a biomarker for schizophrenia and dementia.
So next we'll look at how we analyze in more detail. So welcome to the ADBS Neuroimaging Center, or ANC as we like to call it. So this houses an MRI scanner which is dedicated for the ADBS study. A majority of our study participants, which includes patients, their family and healthy controls, undergo MRI scan right here. MRI suite, you can see that this is the console room where our technicians operate the scanner. And this machine is the Philips Ingenia CX, which is our 3 Tesla MRI scanner. So an MRI scan is a really safe procedure and there's no exposure to any harmful radiation, unlike other scans such as X-rays or CT scans. But the magnet staff will do a safety check with you. Any metallic such as braces for your teeth, heart implants such as pacemakers, or if you have undergone any surgery that involved fixing any rods, screws or clips inside your body. So the next step is also a metal screen, just like what's done in airports, to be doubly sure that you have removed all other metals such as jewelry or electronic gadgets before going in. Up to. So now that he's been positioned nicely and has gone inside the scanner, we start the scan. We'll now show you another short clip which explains the different types of MRI scans acquired in our study and some of our research findings.
This is an example of two distinct types of high-resolution structural images that we get from an MRI scan. Typically speaking, if a physician asks you to get a brain scan, it might look like one of these, although other types of scans are possible. The one on the top is called a T2 scan and the one on the bottom is the T1 scan. If you look carefully, you will see that they are opposites of each other. What is bright in the top image is dark in the bottom image. These T1 and T2 are weightings. Different weightings of the image will give us different information. This is a cartoon representation of a typical neuron, the cells in your nervous system. Note two important parts of this diagram, the gray-colored cell body on one side and the white-colored axon stemming from this body. In the bottom image, the central part of the image is bright. This is the white matter of the brain consisting of the axons. Many axons are covered with myelin, which gives it the white coloration. The thin dark strip on the outside of white matter is the gray matter, which consists of the cell bodies and some other cells. The central dark portion embedded in the white matter are the ventricles, which are filled with cerebrospinal fluid. This is an example of a different type of scan, the functional MRI. This tells us which parts of the brain are engaged in an activity at a given time. This short clip shows the fluctuations in the MRI signal over time. Functional scans are typically lower resolution. Notice how the brain looks blockier here. On the right-hand side is an example of the default mode network. The DMN is a network of different brain regions interacting with one another when a person is at rest. That means, not focused on anything in particular. These are derived after processing the raw data. The final type of example we will show you today is the diffusion-weighted imaging. This type of scan is focused on the white matter, in which we track how the water molecule is diffusing along the white matter of the brain. The clip on the top shows the raw data, while the bottom image shows the white matter derived after processing. We follow strict and rigorous quality assurance and quality check protocols to make sure that the data we acquire is of good quality. One of the important parts of this protocol is to perform daily and weekly phantom runs. MRI phantoms are MRI-safe objects with different properties which behave in a particular manner. Here we show examples of three types of phantoms. MRI phantoms, just like humans, come in different shapes and sizes. Using these phantoms, we can ensure that the MRI scanner is calibrated and everything is working as expected before your scan.
Now, the blood sample which we collected from our patient is transported to the Neurobiology Research Center. The NBRC building houses the Molecular Genetics Lab and the ADBS cell culture facility. First, the sample goes into the MGL lab, where it will be prepped in order to isolate DNA for further analysis.
Hello, welcome to the Molecular Genetics Lab. This is where we've been studying the genetic basis of neuropsychiatric illness for the past 20 years. Dr. Jain here will explain to you what are all the diseases that we've been worrying about over the years.
So the common severe mental disorders like schizophrenia, bipolar disease, addiction, OCD, dementia often runs in families, and these are severe disorders. So we try to collect the DNA, we try to do some DNA analysis, and Dr. Mira will run you through that. But as we know, these diseases are mainly expressed through the brain, and since we can't study the brain, we've also added a stem cell section where we try to grow brain cells from patients who have these. We can combine the genetics with the biology and try to gain new insights into how the disease occurs and why it progresses the way it does and how it responds to treatment.
So, as you all know, DNA is the blueprint of the cell, and the most easiest source that you can get of DNA is the blood. And so the blood sample that is collected from the patients is then analyzed here in this laboratory. As a first step, we isolate cells from the blood and we go on and isolate DNA from that. Further analysis tells us what is the difference between patients and unaffected people in the DNA.
DNA is like a cookbook for your body. It contains recipes or codes to make different proteins which are essential for the survival of an organism. When this DNA divides, The recipe is prone to changes, which are called mutations. Usually, these mutations do not make a difference, and we often don't even know that we have them. But sometimes they can be harmful and lead to diseases, including psychiatric disorders. In our lab, we study the changes in the DNA sequence to learn how they might cause different disorders. We sequence the DNA to figure out what these mutations are, and then carry out several kinds of experiments to understand how they contribute to these diseases. As you can see in the red circle, this is the isolated DNA from the blood samples that we get. Once we finally have access to the DNA, we need to make sure that it is pure and intact. To get an idea of the quality and quantity of the DNA that we just isolated, we carry out a process called gel electrophoresis. Gel electrophoresis gives us an idea of the quality of the isolated DNA and shows us whether it is intact or is fragmented. A single and clear, sharp band on the gel indicates that the isolated DNA is intact, whereas a smear on the gel shows us that the isolated DNA is fragmented and thus cannot be used for further analysis.
So here in the ADBS lab, we get blood samples from everyone who agrees to participate in the ADBS project. From those white blood cells, we generate what's called lymphoblastoid cell lines, or LCLs. They look like this. This is so that we can have a permanent repository of everyone who participates in the project. And so from these cells, we send them to our collaborating partners in NCBS and InStem, where they generate stem cells. This is a technology that allows us to take any cells from the body, blood cells, skin cells, and generate what's called pluripotent stem cells. These are the cells which create all the cells in the body. So once we get the stem cells, or iPSCs, induced pluripotent stem cells, back from NCBS, then do further studies here. That is to try to understand how the brain cells in patients that have different their brain cells compared to others who do not. So here today we're going to show you snapshots of what those cells look like and how we use the different experiments we do to try to understand their differences.
Now Swarupa is taking the blood sample into the ADBS cell culture facility. The cell culture facility is a sterile place and needs to be free of any contaminants. Right now she's entering the air shower, which will remove any particulate matter and keep everything clean. This is our lab technician Karthik. What he's doing right now is carefully layering the blood on top of a solution in special tubes, which help separate white blood cells from the rest of the blood. After we get these white blood cells, it's time to count them so we know how many cells we get. We do this using a device called a Hemocytometer. These tiny white specks that you see inside the squares are actually cells, and we're able to get millions and millions of cells from this small volume of blood. Now we turn these WBCs, or white blood cells, into lymphoblastoid cells. Scientists like Vasvi will then grow these cells and make sure that they're healthy. Let's take a look at them. These cells stick together and form circular clumps. Now those definitely look like some happy cells. Now, these LCLs are turned into iPSCs, or induced pluripotent stem cells, by our friends at InStem and NCBS. Stem cells are basically the raw material of the body, and all other cells are made from them. Scientists figure out a way to turn back the clock into pluripotent. Do this in a lab. This incredible discovery won them the Nobel Prize in 2006. These stem cells are specific to the brain and will eventually grow into different kinds of brain cells like neurons, astrocytes, and oligodendrocytes. Finally, These neural stem cells are grown into small 3D tissue cultures called organoids. Organoids are a simplified and miniature version of an organ, which in our case is a simplified brain. The first step to creating an organoid is making a 3D spherical tissue culture called a spheroid. Now that we've got our brain cells ready, it's time to identify them, the same way that an ID card would identify me or you. We use a technique called immunocytochemistry or immunostaining to identify which specific cell we have with us. Let's see what Srividya is up to. She has grown some cells on these delicate glass cover slips placed in this small plastic dish. She's carefully gone through multiple steps of an immunocytochemistry protocol in order to detect and visualize specific proteins in her cells to be able to identify them. She's transferred the cover slips to a glass slide in order to view the cells using a fluorescent microscope. Let's see what her cells look like. This is a cortical neuron. The red color that you see is a marker which is specific to a certain protein inside these cells that help us identify them. Now this is the green color is specific to a certain protein in this cell, and it helps us identify a neural stem cell. The reason that they are different colors is because these different proteins will help us identify which cell is which. Since these cells come from patients with various psychiatric disorders, they may behave differently from normal cells. We use these cells to do experiments in order to understand these differences. For example, we compare the movement of cells which come from an unaffected person, or what we call in the lab a control sample, to cells which come from a patient. This experiment is called a migration assay. These are some neural stem cells which we grew in a small petri dish and then looked at under the microscope. You can see them moving around. Pretty cool, right? Now, let's compare cells from an unaffected person, or what we call a control line, to that of a patient. What you're seeing now are the control cells moving. And these are how patient cells move. Can you see a difference in the movement? This is just one example of several differences we see between control and patient cells.
Hi, I am Yojit, a PhD student in Professor Raghu's lab here in NCBS. So far, you've heard my colleagues at NIMHANS describe their workflow and explain to you what stem cells are and how one can identify cells of the brain. I, on the other hand, will take you through one special aspect of neurons, and that is how do the neurons communicate with each other. So, come with me to ADBS lab here in NCBS to see how this happens. Understanding how neurons talk to each other is important, because in the human brain, neurons process information and communicate with other neurons and finally with the rest of the body. This way, the neurons instruct the human behavior. But performing any kind of brain function requires many neurons in different parts of the brain to communicate with each other. Mutations in genes can disrupt this very communication. Thus, it becomes useful to assess how neurons are communicating. They achieve this communication by sending electrical signals, which triggers an influx of calcium ions into neurons. Using dyes that bind specifically to calcium, we can monitor neural activity in neurons. Accordingly, in ADBS NCBS, we have set up a technique called calcium imaging that can detect changes in the calcium ions. Here is a dish which has neurons derived from the human stem cells, and these neurons are loaded with fluorescent dye that specifically binds to calcium. The simplest way to detect fluorescent signals is through this microscope called as the wide field microscope. In order to check for neural activity, we need to carry out time-lapse imaging in the dark. The blue light that you see here excites the fluorescent molecules that are loaded in the neurons, and these in turn get captured by the camera and can also be seen here on the screen. I just imaged these neurons, which were maintained for 1 month. The crosstalk between the neurons can be seen through the flickering of activity that you see here. These are calcium ions that are either going inside or outside the cells as instructed by the neurons. Scientists use this technique to understand how neurons interact in the brain, as well as understand how neurons from patients with various psychiatric disorders behave differently from the normal cells.
So that was the video. And so moving forward, so we just seen what the ADBS program does and what are the various aspects of the illness which is getting covered and what are the various brain measures. So what we managed to do in the last few years is to screen around 5,000 families and do detailed assessments of more than 700 families across the country. And yes, these are not just resources created for this group, but this is supposed to be an open repository where any scientist can ask for resources and start working on the biology of brain illnesses. In terms of future of the program, there are immediate goals and long-term goals. One can look at emerging patterns, and this is something which is ongoing work, but going on to the future. And emerging patterns from multimodal data sets to try and understand the diagnosis better, to make better diagnostic classifications, do early diagnosis because we are looking at patients who are not yet developed the illness with the families. In terms of personalized medicine solutions, try and understand who responds to treatment, who will not respond to treatment, and why is it so. And of course, develop new treatments because these are human systems, and experiments done on human systems, compounds which have a less failure rate in human beings. So with that, I would thank the team. I'm the person who has done the least work for this program, and all the work has been done by the team. And I think starting from the left, Anushka, Srinivas, and Soumya have been the masterminds, and they've done everything for the project. Soumya was 9 months pregnant when she videoed all this. It's just too much commitment, and I can't say how much I'm thankful to them. The other team members, Yojit, Nirmala, Pravesh, all of them were speakers. The senior team members who helped them immensely, Raghu, Meera, Ritika, Dr. John. Of course, the program would not have happened without Sanjeev. Video credits to Daksh and the Mental Health Education Department at NIMHANS. A lot of team members who helped at various time points, and they are I can't name all of them. There are some 40 members who participated. Many of them, were acting as patients. Many of them volunteered for a lot of background work. So thank all of them. And of course, funding by the DBT and the Pratiksha Trust. But then also the video was funded by the India Alliance, which also funds me for my interpretive fellowship. Thank you so much, and we will take it up with Madhu.
Thank you, Biju. If I could now invite all the experts who will be a part of the Q&A session to have their cameras on, so we could see them. And I'd also like to encourage the participants to share their questions in the Q&A box. So now we'll have a discussion with the researchers from NIMHANS and NCBS about their work. So if you have any questions from the walkthrough, please do add it in the Q&A box. There is already one question in the chat box, but if there are others, please do share them. I'm sure all the experts here will be very happy to take them. So Biju, what I'll do is I will share the question, and you can maybe suggest who would be best equipped to answer it, or if all of you would like to start off. So the first question that came in while the walkthrough was on was a question about the LCLs that you guys are working with. So it's a question from Saurabh, and he's asked, what markers do you use for LCL characterization apart from the way they look, the morphology?
Yeah, so just similar to all the other cells, there are immunophenotypic, immune markers which are available for all of these. So there are markers to make sure that these are B cells. These are supposed to be B cell lymphocytes and lymphoblastoid. So, and you make sure that they are negative for the other cell types, the T cells. So I don't want to go into the exact details of what it is in this probably, but actually we have specific markers for each of the cell types. So that is also done in addition to morphology and all the other characteristics.
Okay.
Yeah.
Thank you. While we wait for the audience to add the rest of the questions to the Q&A box, maybe I can get us started by asking one sort of broader question about the ADBS project. So would you like to, for instance, share some of your key findings and biggest challenges, given that this is such a broad project that that your walkthrough showed, it encompasses many kinds of techniques, many sort of specialized disciplines and methods of study within this program, and you're working with everyone from people to the most fundamental cells. So some of your most interesting findings and the biggest challenges that you faced with this project.
I mean, I will request the senior most members, Professor Jain, Raghu, and Meera, to respond to this separately. So I think that should cover many things. Professor Jain or Raghu, whoever can start. Okay, or Meera, I don't know who's there. I thought all of them were there.
Yeah, so
Yeah, Meera.
Okay, maybe I can start.
Yeah, so the challenges wise, yeah. So if I say what are the things that you found to start with, so the reason, one of the primary reasons that we started the study was to test out the cross disorder hypothesis, and indeed for family histories of pedigrees, we found that there were many families where there were different kinds of psychiatric illnesses that were found together. Like you would have even the pedigree that was discussed, for instance, in the initial generation there was dementia, and then there was bipolar disorder, and then we would find somebody with OCD. So if it is genetic, there is a lot of cross heritability as well, and that is something that we have gone further, and we've published a few papers doing the exome analysis data. So it is becoming increasingly clear now that there may be the same gene being affected, having mutations in different parts of the gene and different kinds of mutations, or a combination of different mutations can give rise to different disorders, even though it's the same gene. So it's not like you have a gene that is restricted only to schizophrenia or whatever. So because finally you're perturbing neuronal function, and depending on the kind of perturbation in a particular individual, you may see a different clinical phenotype. And the other, one of the interesting findings that we found, which was also shown, was the migration assay. We could definitely see that there is a difference. There seems to be a difference in the migration of an unaffected individual versus an affected individual. So that's really something quite amazing, because if you think about it, you've taken blood cells from both these individuals and you've differentiated it to make neurons, and they are now behaving differently in the dish. So it sort of gives you confidence that yes, once you have a model system that you try out, ask different questions you want to look at drug treatment or stress or whatever.
I think Raghu was.
You're muted, I think.
I think Raghu was trying.
I've seen a large number of different types of measurements that report how the brain of a person is working at different levels, starting from the output of the brain, which is behavior, through to the structure and function as measured by varying techniques. And then towards the end, you also saw experiments that are trying to recapitulate in dish in the laboratory how the cells of a brain cells of a techniques reporting brain activity at different degrees of resolution and scale. And I think that the big challenge for scientists, not only us, but also all over the world, type measurement, other levels, and finally related to one of the important reasons why we are doing all of this. There are individuals who are ill, mental illness or mental disorder, and we'd like to understand and find ways of helping. This remains at the moment largely a laboratory venture. It's important to understand that to put anything back into a human patient, one must have a high level of understanding of the consequences of that will arise. And we don't know enough about that in order to be able to do that. Now, there are, of course, certain brain disorders, such as Parkinson's disease, where specific cell types are missing from the brain. And those cell types, when they're missing, give rise to the brain disorder because they produce an important chemical, in that case, dopamine. So that is a setting in which I imagine sometime in the future, we will be able to use stem cells to recreate the neurons that normally produce dopamine. And I can envisage that happening at some point when all safety and other considerations have been met. But, as Sanjeev, I think, mentioned a little while ago, finally, the human brain that we see in an adult is the fruit of development of a lot of connections being formed between various cells in the brain. And that all takes place during development. And at the moment, it's a little difficult to imagine how you could put in adult stem cells into an adult brain and expect that to be recapitulated in the adult. But you never know. I mean, I think probably even 10 or 15 years ago, I wouldn't have imagined that some of the things that we are doing today, even in the lab, were possible. So I wouldn't say it's not possible, but it's some time away.
Thank you, Raghu. The next question that has come is from Parth, and his question is, what are some of the translational application possibilities from the data that comes from the research carried out by your group? Biju?
I'm just seeing if there is someone else who
would like to take this.
Who is not spoken on this. Right. So, I mean, in terms of some of the interests that I have directly been part of, I mean, we are trying to see, we have patients who are given psychiatric drugs, and people do get better, but then we don't know who responds to which drug. It's all a trial and error. People are given drugs thinking that they will respond. We have to wait for like months or years to know whether they will respond. And if they don't respond, then they put on another drug. So it's like we sit and wait to see whether people get better. Now, is there a way that in the lab, we'll be able to pick up whether, with an experiment to figure out whether a person will respond to a particular drug? And that would have, I think, an easier translational potential than anything else. Of course, I mean, if you find new pathways, then you can think of new drugs, whatever, but that's some time away. But that will be something that I can think of immediately. That would be very important. Yes. Now, is there anything more that anyone wants to add? I'm happy to.
Yeah, I mean, maybe I can add a few words. So as a clinician, when somebody comes to you with an illness, be it a brain disorder or something else, even before we get to the treatment, I think the first point that we'd all like to be able to do quite effectively is to diagnose the illness accurately. And I think it's been mentioned a while ago that many forms of mental illness are, they're related to each other. It's a spectrum of disorders with overlapping signs and symptoms. And some of the work that is being done in ADBS will ultimately lead to more accurate and effective assessment of the patient when they first turn up at the doctor's clinic. The second point about mental illness in particular is that these disorders often run for many years and are coincident with is usually seen in young people when they're making many important transitions in their life and careers, making decisions and so on. When there's an illness that runs for such a long period of time, different patients turn out in different ways. Some will get better, others will take much longer to get better and so on. And so as a physician, one would like to have some idea of what the trajectory or the clinical trajectory of a patient is. And as it was mentioned in the start, The ADBS program it's a prospective study, so patients are going to be followed over a period of time. Now you might wonder why one wants to do that, and one reason for doing it is that it's possible to monitor the way the illness develops in this group of patients. And one very obvious translational outcome of that is that the physicians and scientists working in the program will then be able to derive from that what might be some of the earlier signs of illness before patient is very severely ill and comes to you in the clinic. So this again will contribute to a better understanding of how to diagnose the illness and also to predict how the illness will progress and for the treating physicians to plan their management of that patient better.
And to carry on from that, I'll just take a minute. A lot of it is the symptoms of illness are one thing, but the basic biology of the illness is something else altogether. So as we do this prospective study, which Raghu just mentioned, you can also figure out whether the brain changes occur, how much do the brain changes correspond with any other symptoms, and what is the essential things that we need to look out for that will alter the condition of the patient for the better or for the worse. So it's not just, so we may be able to discover things, for example, like exercise or lifestyle or the absence of stress, which actually produces a biological effect on the brain. So lots of these things can be understood in a very dynamic way. And the environment can also be therefore evaluated in its effects on recovery, resilience, and restitution in the brain.
And if I may add a small point, that mental illness is always linked with so much of social stigma. So if you're able to better explain why something is happening, for instance, you know what's going on in diabetes. So, people find it easier to accept that, okay, this is the problem here. But mental illness is still sort of, it doesn't really boil down to a gene or a biological pathway as yet, though it is there in the papers. The social acceptance of it is still, there's a lot of hesitation to, just call it a disease of the brain. It's something, it has a very nebulous social implication. So if you understand the disease better, probably some of that at least will fall away and patients will be treated with more respect and they can be also, come for clinical help. That's what
I think the minimum time gap between, treatment, the treatment gap, I think is 6 or 7 years before someone comes to the doctor. That is what stigma does to, illness, because people don't get treated because nobody wants to be known as ill, as mentally ill. That's a big problem. So yeah, I think Mira's point is very, very important.
Thank you. So there's another question which I would like you guys to take before we come to a series of some common questions. This is a question from Dr. Abhijit Banerjee, and his question is, are you studying any epigenetic mechanisms which could be modulated in potentially affected families?
I'll give it over to Mira.
Yeah. So one of the big problems of studying epigenetics in a human actual life context is that every human being's social context is different. The social stressors that they go through is very different. So it's a very difficult experiment to do, but that doesn't mean that we are not trying to do it. It's for example, we have a bunch of twin samples where we want to see, even monozygotic twins who have the same genetic material. One twin gets ill and the other twin is fine. So that's an ideal situation where you can actually look to see whether there are any epigenetic changes, which is the effect of stress. So we've done other studies where we've looked at stress in children, like children of alcoholics versus regular children, and we found that there are epigenetic marks in children. Similarly, we looked at epigenetics in alcohol abuse and found that there are epigenetic marks. So yeah, we are definitely looking at that as well. But they are difficult experiments and they take a very long time to, make strong conclusions. But we have not given up. We are on it. Yeah.
Which is true for most of this, I think.
And also, another interesting experiment that we have done is looked at epigenetic signatures in the developing brain, the postmortem brain samples, and we found that the methylation marks change over time at different ages. There are different genes are methylated and not. So there is a lot to be understood with respect to epigenetics and the functioning of the brain for sure. Yes.
Specifically in the ADBS program, because of the follow-up study, we do take repeat samples of DNA. So looking at DNA
So over
methylation
time, we
and
can study the epigenetic signatures and see how it
yes,
changes.
yes. So that's definitely in the plan.
Thank you, Meera and Biju. The next question I'll get. So there are two questions which are somewhat similar, and the participants wanted to know how do they, what kind of educational background is needed to come be a part of a team like this or to even intern with you guys. And there's also someone who said that they're from a microbiology background, but they're interested in working with stem cells. So maybe you could talk a bit about, what it takes to be a part of this team and, conduct research of this sort in terms of disciplinary.
That was a very, very, very long answer. So how do I cut it short? Yeah. So essentially, we are. I mean, as we would see, we start from clinic. We do stuff, at the human brain level. We are doing MRIs, EEGs, wet lab work, DNA work. So I mean, this will include all kinds of people from medical background, psychology background, technical background, technicians, life science background. So yeah, it's very broad. And we will have ads out every month. I think we have some ad or the other out every month. So, and of course, you can just write to one of us and we'll see what we can do. I mean, there are various parts, so many parts of the program that I'm sure some people can be plugged in. I mean, it's been a little difficult with COVID and all that, but yeah, I'm sure there are ways to go about it. I don't know if I answered the question. I don't know if anyone else wants to give a better answer, but yeah, it's just broad.
Just write to us if you're curious. We'll find some way to use your curiosity.
Yeah, that's what it is. Yeah.
Thank you. Okay, there's another question. So it's sort of a two-parter. I'm going to try to put it together. Is there any in silico platforms to explore more information specific to neurodegenerative disorders? If you're interested to get into, say, drug repurposing or drug design.
Okay. I mean, we are not, at least in this program, we are not having in silico platforms. There are so many, I think. Meera, would you know anything? I mean, there are so many platforms available for this kind of work.
Yeah. So, first of all, there's a lot of, for instance, when let's say we find a mutation or a variant, right, in a gene. So, is it a variant of major effect? So how do you find that out? You try to put that variant in the context of the protein and see, is it going to change the structure of the protein or not? And let's say that particular variant is very close to the active site of an enzyme. Then you would expect the activity of that protein to change as a result of this particular variant. So, and then to take it further, you could of course do drug binding studies and all of that. So yeah, definitely there is a place there to look at not only dementia, for that's true of any exome data that you might get. If you look at the list of deleterious variants that you get and then go back and put that into the protein model and then look at drug binding studies, yeah, that's definitely something that's doable, and that's also something that we are doing on the side in the lab right now. Not maybe as a main focus of the ADBS program, but we are also trying to do this activity.
And I think there are lots of programs which are doing it around the world, and most of them have data which is openly available. Open data has come up in the last few years.
AlphaFold, for example, was a big initiative where now anybody can look at structures. Earlier, you had to have first a crystallographic structure with which you had to work, and there were only packages like Schrodinger, which was a commercial package, and therefore you had to, which is paid, and therefore you have to be a part of a big lab and
sort of a standard venture where we can do this, but now you can do it as a cottage industry because of AlphaFold, and you can play around with structures and see what happens when you make changes with the amino acid sequences. So there are, maybe when you have to do it more seriously, you have to get into the nitty-gritty, but as for a start, you can definitely play around with things like AlphaFold to start with.
Thank you, Meera. Is there anything else anyone would like to add? Otherwise, we can go to the next question.
No, that's fine, I think.
Okay. So the next question from one of our participants is this: is a patient suffering from a psychiatric disorder predisposed to other multifactorial disorders such as diabetes? Is there something that you guys have seen from your studies, or is this something that's well known?
Well, I'll hand it over to Dr. Jain. If you...
And I'll try to be as brief as possible. I'm running low on battery. So many complex diseases do share overlaps, and am I audible now?
Yes. Yes.
Yeah. Okay. Yeah. So many disorders do, like patients with schizophrenia have a 5 or 6 times higher risk of diabetes. Patients with bipolar disorder often develop cardiovascular disease and often have almost a 3 to 4 times higher risk of hypertension and dying of a heart attack. But since those disorders begin in the 40s and 50s, we don't have enough long-term data on patients to see what actually is the shared biology. So that's another area that the ADBS will fill up, because in the family that we see also a clustering of diabetes, hypertension, thyroid disease, then we know that there are links between these disorders. So those are some of the other spin-offs that we hope to get over the next few years.
Thank you, Sanjeev. There's still one more question. And if there are any more questions, I would encourage the participants to add it now, because I think we will be wrapping up soon. This question is, because stem cell based therapies are at a very nascent stage in our country, what is your take on its future in terms of with respect to bioethics? Do you as a team have any thoughts on this?
Raghu or Ritika, you both.
Well, I think that's a very large question, but I will try to say some things about it. It's true that stem cell therapies are at an early stage in India, but there is also, and perhaps the ones that are likely to come online and be available, and are already, some of them are already available, are in relation to disorders of the blood or hematological disorders, where it is possible to take out stem cells from the bone marrow, modify them as required, and put them back. There are also other developments afoot in different parts of the world to try and use themselves to treat some aspects of diseases of the nervous system. The one that's most likely to work first is treatments in the retina, where one can replace certain cell types when they undergo retinal degeneration in diseases such as retinitis pigmentosa. I think that one big challenge in using stem cells to treat diseases is that it is important that we, if not absolutely assured, that the treatment is unlikely to cause any harm, and second, that it's going to have a benefit. While there is a lot of discussion about technologies such as genome editing and stem cells, there's still some distance to go before one can be assured that editing and so on don't leave their marks on the genome in a manner that their use in therapy will result in some other undesirable or unexpected outcome. So I think it's very important to discuss these ethical considerations, and we do need to keep doing that now, even though the treatments themselves may be some years away for the other disorders. There are there is a part of the stem cell program not directly related to ADBS that is dealing with a very practical need in patient care, and that is the need for transplantation. Now, these days, if you need a transplant of any sort, one of the big challenges is to find a matched donor of the right haplotype so that the organ that is transplanted is not rejected. But just like the brain, what you've seen in the case of the brain today, there are equally exciting developments about the use of stem cells to produce tissues and other tissues and organs, or muscle, parts of the liver, and so on. Now, all of these things, of course, are examples of replacements. So when something is damaged, you replace it with the tissue derived from. Possible in the area of stem cells, because if you can find a donor, or you yourself can donate some cells to generate stem cells, it's possible to regrow skin or some other tissue that you need. But there are still considerations about the experimental procedures that are used in the lab prior to using them in patients, and these are being dealt with at the moment. But haplobanking, which is the science of producing a bank of stem cells, which will fit the needs of most human beings across the world from a relatively small number of donors, is in an advanced state of development. It has been in different parts of the world for quite some time. But the ASHD program, of which ADBS is a part, is also building a haplobank of stem cell lines from which it may become possible to produce tissues for use in human disease therapy. This program is based in the Christian Medical College in Vellore.
Raghu, there's just a small follow-up to that question itself from another attendee who said, who sort of says that their question might be slightly irrelevant or futuristic, but I'm not sure it is. They ask, how far are we from developing organs from stem cells? If you could just maybe respond to that in continuation of your previous answer.
I said if you're talking about it as a principality, this is already done. People are able to isolate stem cells and put them back into the patient and repopulate certain specific types of blood cells, for example. It is also used in some places to try and grow sheets of skin for transplantation and so on. So that is possible. But as I said, for whether it's stem cells or any other form of treatment, developing in the lab is an essential and exciting part of it. And then there is the next step, which is to ensure that whatever you have developed is safe for use in human patients. And that often takes an equally long time to get through so that patients can then benefit from it. Sanjeev, would you like to say something about this matter?
Sanjeev, you're on mute.
You're muted.
I don't know if his battery is. Oh.
Okay. So the issue with stem cell, especially of the brain, was that in the initial excitement when they did stem cell replacement for Parkinson's disease, many of them developed symptoms of increased dopamine production, hallucination and delusion, because we had no way of regulating the behavior of the stem cells once they were transplanted. And as Raghu pointed out, the feasibility of fine-tuning these stem cell implants to match the developmental trajectory of the brain and the wiring that is appropriate at that point in time is still some time away. So at this point in time, the things will be limited. However, for tumors of the brain, stem cell therapies are being initiated, and make sure that certain kinds of tumors can be handled in a sense by replacing certain kinds of clonal cell lines with some other cell lines, so the tumor literally is phased out in a sense. But these basically require surgeries to implant the stem cells back into the brain. The hope is that eventually, when all these technologies develop, one should be able to fine-tune the implantation of stem cells also into particular circuits in the brain, and perhaps do that better. But those are really futuristic technologies. As far as organ systems go, Raghu's already mentioned synthetic skin. People are trying to grow synthetic valves, blood vessels, liver tissue. So things like that are being attempted, and they may actually be feasible in the not too distant future.
Thank you, Sanjeev. Thank you, Raghu. There are two more questions which we'll take, and then I think we will bring the session to a close. The first question is, are there any systemic mechanistic links between neuropsychiatric illnesses and neurodegenerative disorders at a molecular level?
Yeah, I think it'll go back to Sanjeev probably. But yeah, we do find that there are multiple. I mean, that's again something that we have been discussing, that similar processes are implicated in many illnesses, and not just adult psychiatric illnesses, but also dementias, autism, which are very, very, very different extremes. But yeah, Sanjeev, you want to add something?
Yeah. So this is something that we are specifically been interested in. For example, patients with autosomal dominant disorders like Huntington disease develop neuropsychiatric deficits, which often individuals themselves are not aware of, decades before the symptoms develop, and 20, 30 years before the symptoms develop. So neuropsychiatric changes may precede the so-called evident physical effects by many, many years, even for known genetic disorders. So obviously, and even with things like ApoE4 and dementia, there is some interest in seeing how genetic variations actually correlate with neuropsychiatric traits. Now that's obviously an interesting question, but at the moment the answers are really metaphorical, because all of us will carry genes for many diseases. We must understand that too. Each one of us will carry hundreds of genes which predispose to some brain disease or the other. So correlating these will really be a matter of time. And in the West, the parts of the UK Biobank and behavioral genetics does just this. But whether they actually correlate to disease in the future is something that will emerge only in the future.
Thank you, Sanjeev. The last question is from Manuel, and his question is, have you engaged in organs on a chip approaches to study complex relationships like the blood-brain barrier?
Well, the simple answer is no, unless Raghu has something to add to it. But yes, that is something that, yes, Sanjeev.
So there are a number of companies that do this, and we did because Bangalore is an IT sector, so there were a number of specialist companies which had developed neuron on a chip, infections on a chip, cancer on a chip, but most of them are Indian startups, and many of them have now migrated to the US. So that's the long and short of it. So there is a lot of interest and technical capacity in India to do this, but a lot of this is part of the global IT stuff. But there are people here who do this. I don't think we have ever specifically engaged with those kinds of efforts, but there are people who do this in Bangalore. Raghu, I think in the CC camp there were certain people who were doing that.
Raghu, is there anything you'd like to add?
No. I think it's been answered at the level that it's possible to answer it right now.
All right. Okay, I think this is a good point for us to bring the session to a close. Thank you so much, Bidu, Meera, Raghu, Sanjeev, Srinivas, Ritika, Yojit, Anushka, who all sort of made it here and were very happy to take so many questions from our audience. I'd like to just tell the audience that if you want to watch this session again, if you feel like you missed something in the walkthrough, you want to go back and check it out, the recording of the event will be on our YouTube channel. And also, if you missed some of our previous events, you can check them out on our YouTube channel as well. One event that I might particularly recommend is the panel discussion in which Raghu was in conversation with Sridhar Venkatapuram and Sujata Raman, titled Thinking in a Dish, about stem cell research and the human brain. So if you are interested in the ethics question, you can have a look at that session where they dove deeper into that topic. You could also see a very interesting exhibit which was developed with the NIMHANS team, which is called Schizophrenia and the Brain. So this is from work from Dr. Urvaksh Mehta's lab, which looks at the DMN, which was mentioned briefly during the walkthrough, and its relation to schizophrenia. So you can check that out in the exhibition, and that will be available to view today. I'd also like to remind the audience again to share their thoughts about today's session in the feedback form. Tell us how you enjoyed it and what else you'd like to see from us going forward. Do see the exhibition today before it closes. And thank you all so much again for joining us on this Sunday afternoon. Again, thank you so much to the NIMHANS NCPS ADBS team for being here and taking us through your experimental spaces, your work, your research, and engaging with the public today. Thank you all.